Thank you, Professor Ellen Kumaat, for officially opening the Icon Smart 2021 and a remarkable speech. Ladies and gentlemen, this concludes our opening ceremony for the Icon Smart 2021. And now we may continue with the plenary session, who will be led by Megumi Najoan, Bachelor of Science, graduate of Faculty of Mathematics and Natural Sciences, Ratulangi University, Manado, Indonesia. Ms. Megumi Najoan, please.
Thank you, Cheisy. Good morning and welcome to plenary session of the Second International Conference on Natural Sciences, Mathematics, Applications, Research and Technology, or ICON SMART 2021. Ladies and gentlemen, I am Megumi Najoan, the Master of Ceremony of this session. I'm very happy to see you here and welcome all of you to this plenary session of ICON SMART 2021. Our plenary session today is about material science and bioinformatics for medical, food, and marine industries. Ladies and gentlemen, this plenary session will be divided into two parts. The first session will be moderated by Feki Mantiri, Ph.D., and the second session will be moderated by Dr. Maria Cristina Tapilo. And before we begin the session, for all the participants in the Zoom meeting room, please change your name with the format class_your name. For an example, as shown on your screen right now, G_your name. And furthermore, at this Zoom meeting, there will be a selection of the best presentation in each class. For that, please all participants giving your best at the section. And now we begin the plenary session that will be led by a moderator, Mr. Feki Mantiri. Feki Reki Mantiri, Ph.D. is a senior lecturer and the Vice Dean for Academic and Collaboration Affairs of the Faculty of Mathematics and Natural Sciences at Ratulangi University, Manado, Indonesia. He holds a Ph.D. in Biological Sciences from the University of Newcastle, Australia, and Master of Science in Molecular Biology and Biochemistry from Simon Fraser University, Canada. His fields and expertise and research interests include molecular biology, environmental biotechnology, and bioinformatics. Well, Mr. Feki Mantiri, Ph.D., the floor is yours.
Thank you very much for the introduction, Megumi. Good morning, everyone. Welcome again to the plenary session of this year's International Conference on Natural Sciences, Mathematics, Applications, Research and Technology, or we abbreviated ICON SMART. My name is Feki Mantiri. I am from Faculty of Mathematics and Natural Sciences, Ratulangi University, and I have been given a daunting task to chair this plenary session. Well, I hope you have enjoyed the conference so far, and let's now proceed with the presentations by the plenary speakers, which I hope you will enjoy equally, if not even more, because we have some interesting talks upcoming. Before we get started, I would like to go over a few items so you know how to participate in this plenary session. This online conference is being recorded and is streamed live on YouTube. We will be able to share a link with you after the event is complete, and we welcome you to revisit the content yourself and share it with your colleagues. While the presenters are delivering their talks, participants will have the opportunity to text questions to today's presenters by typing your questions into the chat area or the chat box. Please write down your question and to whom your question is addressed to. We will then choose the questions that will be discussed during the Q&A session. In this plenary session, we will be listening to the presentations from 5 speakers from different backgrounds, either as academics, researchers, or practitioners, professionals. They are Professor Dr. Michael Knoblauch from Washington State University, USA, Mr. Abrizal Andrew Ang from PT Samudra Mandiri Sentosa Bitung, Indonesia. Mr. J. Irwan Hidayat from PT Industri Jamu and Pharmacy Sidomuncul Tbk Semarang, Indonesia. Professor Dr. John Wang from National University of Singapore. Professor Brian Yuliarto, Ph.D. from Institut Teknologi Bandung, Indonesia. So let's get started with the first speaker, Professor Dr. Michael Knoblauch. I believe Professor Knoblauch is already here. Okay. Good morning from Manado.
Hello.
Professor Michael, you still remember me? We had some session last year.
Yeah, 2 years ago I was in Manado.
Oh, 2 years ago, yeah.
Professor Julius Ponto.
Yeah, okay.
And,
so how's everything in Pullman?
well, so far so good. Of course, COVID-19 had a big impact on, as you know, everywhere. So we are slowly coming back to normal operation, step by step. Classes are in person again, and I hope that we get it under control soon.
Okay, so before we proceed with the presentation, let me first introduce Professor Dr. Michael Knoblauch. Dr. Michael Knoblauch is a professor at School of Biological Sciences and director of Francesi Microscopy and Imaging Center, Washington State University, USA. He received his Ph.D. from Justus Liebig University, Gießen, Germany. His research interests include cell biological phenomena that have an impact on whole plant physiology and ultimately on food security, which coincide with the theme of our conference, of course, climate change and bioenergy crops. His current projects include proteomics of sieves elements, phloem unloading, and many other interesting things such as engineering of plants for phytoremediation. So accordingly, today, Professor Michael Knoblauch will present his talk entitled Plant Physiological Strategies to Improve Crop Yield. So ladies and gentlemen, without further ado, please join me to welcome Professor Michael Knoblauch, which will have 20-minute presentation followed by 5 minutes Q&A session. Professor Knoblauch, the floor is all yours.
Thank you very much. I'm going to share my screen, so let me know if you can see it.
Yes, I can see it clearly.
Okay, so I was asked to talk a little bit about plant physiology. I understand that the audience is very diverse, right? I mean, we have people from mathematics, we have people from engineering and so on. So I try to introduce my field a little bit, the background, and then I go a little bit more in detail. I will specifically talk about one project that we do because in 20 minutes I cannot do more than one project. But I think it's one that has specifically to do with food security, and this is one of the topics here, I guess. So let me start. Everybody knows this, right? A standard plant leaf, and the fundamental process of life on Earth takes place in these leaves. This is photosynthesis. Every organism on this planet is dependent on photosynthesis, the conversion of light energy from the sun into chemical energy in form of sugars that the plants consume, that humans consume, that animals consume, that bacteria consume, and so on. So basically everything is dependent on this. And but the interesting thing is we usually don't eat the leaves that generate the photosynthesis, right? Where the sugars are generated. What we eat is this, and these things are called plant sinks. So the leaves here, these are the so-called sources. They generate the sugars. Our food are the plant sinks usually. And these are maybe cereals, grains, and so on, fruits, or the roots, right? Like tubers, potato tubers, carrot roots, and so on and so on. So what we eat is not the leaf that generates the photosynthesis. And somehow the sugars have to come to the sinks, that the sugars that have been produced by photosynthesis, they have to travel to the sinks that we eat, right? And this is what I study in my lab. There is a tissue, a specific tissue, that is responsible for translocating the photosynthesis products into the sinks. And the process itself defines how much a specific sink receives. So at the end, how big our food gets, how nutritious our fruits are, and so on, is defined by the process of the transport and the unloading into the sinks. And this is what we study. So let me briefly show you. Here is an example of a tree, and we have multiple sinks in this tree. So we have the source leaves here that make the photosynthesis. They produce the sugars, and then the sugars can go in very different places. They can go into the tree trunk, so the stem, or the tree branches, because they don't do photosynthesis and they need to be fed like we eat. These tissues eat as well. They eat basically the sugars that are produced by the photosynthesis. The same is true for the apples here. The same is true for the roots of this tree. So this tree has hundreds and hundreds of sinks that all want to have the sugars. What humans want is that this tree produces large fruits, right? As big and nutritious as possible. And we don't care so much about that this tree produces very big roots, very, very big stems. They are not of interest for us. Our interest is to improve the quality of nutrition of the fruits and the amount of the fruits, right? So this is what we did over centuries and centuries via breeding. So we were sitting down and we looked at different trees, offspring of a certain tree, and the one with the biggest fruits we selected, took the offspring of them and looked for bigger fruits, and over time, many years, many decades, the fruits were selected for, or the plants were selected for bigger fruits. With modern technology, we can potentially do better and faster, right? With the current growth of population on the planet and the need for healthy food, no question, we need new strategies, because simply we run out of time. Yeah. If we continue breeding like we did it over the last hundreds of years or so, we will not be able to feed the world population, and we will not be able to provide food that is highly nutritious and healthy. So we need new strategies, and this is what we work on basically. Yeah. So let's go back to the leaf here. The question is, how do plants transport the sugars and other substances like amino acids and so on from the leaves where things are generated to the sinks, into the fruits and roots and so on that we eat? There are two tissues in the vasculature. If you look at the leaf here, this is a leaf plate, and you can see these veins here, these strands, yeah? And they branch out into little, always smaller and smaller branches. And these are actually the veins, and the veins contain two tissues. And the one tissue is a so-called xylem, this here. And the xylem translocates water and minerals from the soil into the leaves. Then the photosynthesis takes place, and the sugars and other substances are generated in the leaf. And then these nutrients are loaded into the phloem, and the phloem, this tissue here, translocates it to the sinks that are of interest for us. So my object of study is the phloem. Okay, I'm studying the phloem, how the substances go from the leaves into the sinks. So what does a phloem look like? I'm a cell biologist. I do a lot of microscopy, and I look at the cellular level at things. So here is a drawing out of a textbook what the phloem looks like. The phloem is a has tube-like cells. So this from here to here is a single cell. And such a cell is called a SIF element. And this cell is connected to the next cell through a so-called SIF plate. So this cell wall here has a big holes inside. And this connects this cell to this cell. And there can be thousands and thousands of cells connected to form an individual tube. And this tube runs from the leaves all the way into the roots, all the way into the fruits, and so on. So it's a tube like a garden hose, just smaller and more complex, right? That connects these two tissues. For many, many years, the hypothesis how this works was proposed in 1930. So some 90 years ago. And it was a long, long question how, actually this is our original book here from 1930, where it was proposed by Ernst Münch. He was a German scientist. And the mechanism he proposed is very plausible, but it was never properly tested. So the mechanism is when you have the source leaves here, this is a sketch, simply this is a cell within a leaf. And the cell makes a photosynthesis and produces the sugars, the little red dots that you see here. Yeah? And these sugars are loaded into this sieve tube, into the tube of the cells that I just showed you. And because of the high concentration of sugars, this cell attracts water. That means water flows in because of the high osmotic potential inside the cell. And these cells, because of that, generate a pressure of about 2 megapascal. That is about 10 times the pressure that is in your car tire. So a car tire is about 0.2 megapascal. These cells generate 2 megapascal. So it's a huge pressure inside these cells. And this pressure, together with some other factors, is able to push the fluid through the tube into the zinc cells. Okay, so all the way from the leaves into the roots or into the fruits or whatever. Yeah, so this is a mechanism behind it. This was proposed again 1930. There was no testing of the hypothesis until about 5 years ago when we did it. And the reason is these cells are extremely difficult to work with. They are extremely small. They're about a 50th of the diameter of a human hair. So extremely small. Because of the high pressure, they are extremely sensitive to injury. So when you want to do microscopy or to investigate them and you cut, if you cut in them, they explode. So what you have to do is to work with whole plants. And this is very difficult to take a whole plant and put it under a microscope, right? And so on and so on. So there were a lot of difficulties. Okay, but what I want to show you is a few results that we got out and that we hope will help us improve the development of plants that generate more nutritious food. So this here, what you see is a root. This is a cross-section of a root tip. This is the outer tip. This here is the root itself. And in this root here, you have these sieve tubes that I just discussed, right? There are tubes in here that you cannot really see because they are too small, but they are present. And I will show you the tubes in a second with a different microscope. But just imagine these tubes come down here, and then the sugars that arrive need to be unloaded. They need to travel from the tube in the neighboring cells so that the cells can be fed. And the question is, how does it work? What is a mechanism to feed all these cells in the sinks? So for this, we developed small boxes. I have one here, right? They look like this. They have a front cover, and you can take this away. And this is what they look like. So this is what you see here. And this is a box where you can fill soil in. And it has in the front a cover slip for the microscope and a meshwork, a very fine meshwork. And when you look with a microscope, this is what you see, right? So you have the soil in the back, you have this meshwork, and then you have a cover slip for the microscope. And between the meshwork and the cover slip, you can put a plant seed. And then the plant grows out. And all the roots are forced to grow along this cover slip here. And then you can simply take this box, put it under the microscope, and you can see the root growing, and you can image it. You can observe what the root is doing, and you can observe what the cells inside the root are doing. Yeah, and you can do that for days if you want to. So this is one development that was important for live observation of the plants. So what I show you now is a movie of a root that is growing in one of these boxes. Okay, and this here is the root. It's done with a confocal microscope. So confocal microscope is a fluorescent microscope, and you use dyes to stain the cells of the plant. So what you see here is simply the root that is stained with a red dye, and these here are individual cells. Okay, and you will see in the movie that this root starts growing. And the second thing what we did is we labeled sugars from the leaves, and we observe how these sugars arrive here in the root. So let me show you the movie. Here you can see how the root is growing, you see? And here, this is a phloem. This is the sieve element, the sieve tube. And here you can observe how the sugars and where the sugars are unloaded. So the green dye, I run the movie again, the green dye that arrives here, this is the transport of the sugar that were produced in the leaves. And then you have the unloading here in these specific zones. So for us, the next question was, how do the sugars or the dyes actually exit the tube system here? When you look at the entire root, the unloading of the phloem only occurs down here in a small group of cells. The root is very long, and all the way up here, there is no unloading, only in this area. And there's a reason for this, because in this area, the root is actually growing, and the sugars are required in this area, and the unloading occurs here. But what specifically defines unloading? How do the cells know that unloading has to occur here, and what structures are responsible for unloading? And this is the next thing we did. So between plant cells, you have connecting channels, and these channels are called Plasmodesmata. So here, this is a word, these are Plasmodesmata. And if you look at the plant cell, this is one cell, this is the other cell, and this here is a cell wall in between. And these connections here, between this cell and this cell, through the cell wall, these things are called Plasmodesmata. Okay? So there is a possibility for cells to exchange fluids. Looking at the movie, however, there are specific cells that receive way more than other cells. So how is this controlled? So what we did is electron microscopy. Here, this is a cross-section of a root. So when you take the root that I had before and you cut it crosswise, this is what you would see. This bright cell here, this is one of the sieve tubes. This here is the other sieve tube. So here in these cells comes the flow and flow, and unloading occurs in the neighboring cells at the specific point. Okay, here is a higher magnification image. So this is one of the tubes here in cross section, and these are the neighboring cells. So we made specific investigations in the cell walls here, and these to see the plasmodesmata there. And in all cell walls that are independent on unloading, where unloading doesn't occur, you see plasmodesmata that look like this. But in the cell walls that are actually unloading, these are the specifically here, you see plasmodesmata that are funnel shaped. They are not narrow like this, they are funnel shaped. Okay, like this. So to get a better idea how effective these plasmodesmata are in connecting cells, we did 3D reconstructions. This is so-called electron tomography. So this is a 3D reconstruction that you can do in an electron microscope. And here you see the cell wall, and here is one of these relatively complex structures, these complex plasmodesmata. It's very difficult to see their structure in a two-dimensional image. Because of that, we do 3D reconstructions to these whole thing. And this is what one of these plasmodesmata looks like in 3D. So you can clearly see here is a very narrow opening, here is a very big opening, and this funnel shape is highly effective in releasing flows from the phloem, from these sieve tubes. Okay, so what did we do next? We evaluated the flow rate through these plasmodesmata. So here's one of these plasmodesmata, and we fed this into COMSOL. COMSOL is a fluid dynamic software that can simulate flows in microfluidic channels. And this is what you see here. So the flow in these white parts there's hardly any resistance. Only in the narrow part here, there's high resistance, and we could quantify the resistance of this. And what comes out is basically that if you compare to a standard plasmodesma, if you have an opening angle of just 1 degree, so this channel here, the yellow channel is a plasmodesma channel. If there's only an opening angle of 1 degree, yeah, like this, from this to this, there's already a massive reduction in resistance. If there's an opening angle of 8 degrees, for example, this is about what standard funnel plasmodesmata look like. The resistance, the hydraulic resistance compared to a standard plasmodesma is just 1.6%. So they are almost 100 times more efficient in unloading than standard plasmodesmata. So what that means is wherever you have funnel plasmodesmata, a lot more of the nutrients will come out. Okay, so what you can do with this is if you can understand how plants control where they put funnel plasmodesmata, or if we decide, or if we find a way to put more funnel plasmodesmata in specific sinks, they will receive more nutrients. So that means if you can control, for example, in an apple tree, to increase the number of funnel plasmodesmata in the apple, then we will get more nutrients in the apple and more yield out of the apple. This is one strategy. Okay. So it is 6:40, I think. Is that?
Yes, you have 2 more minutes.
2 more minutes. Okay. So I will tell you a little bit more about. So what's also important, we need to know the number of these plasmodesmata in order to figure out how we can increase or to screen mutants that might have more plasmodesmata of these or less, right? So this is another very difficult task because these things are so small, you only can see them in an electron microscope. So what we use for this is serial block phase imaging. This is a scanning electron microscope, and basically what it does is it scans the surface of the tissue, then you have a microtome, and the microtome slices a section, and then you scan the next surface. And this way, over a day or so, you can get 500 to 1,000 individual sections. And with this, you can 3D reconstruct. This is a 3D reconstruction of one of these unloading zones with a sieve tube. So this here is one of the sieve tubes. You can see it right here. This is one of the sieve tubes, okay? And now in the cell walls, we can see the plasmodesmata, where they are distributed. And we can extract here. These are all the plasmodesmata. This is the location of the plasmodesmata that we can extract. This is a cell wall, and so on. So this way, with this 3D reconstruction of large scale, of large tissues or so, we can now evaluate how many plasmodesmata are there for unloading, and we can screen when we have plants that are better performing, how many plasmodesmata they have more, and we can have a tool to evaluate, for example, mutants that we generate if they have more of these plasmodesmata to have more unloading in the areas that we are interested in. Okay, I think my time is up.
Yes.
So I'm going to stop sharing and happy to take questions.
Okay. So thank you very much for the interesting presentation. So, well, I haven't seen any question being posted in the chat room, so I will give the chance for the participants to directly ask questions to Professor Knoblauch. Before the participants doing that,
There's actually a question. I see a question here. Do all species work as you described?
Okay.
So we recently have just published a paper where we screened another 7 species from various plant families. And in all species, we find these funnel plasmodesmata specifically in the unloading zones of roots. So they are present. It's a general mechanism, it looks like, yes.
Okay. Okay, since there's no more question so far, so I have a question on my own because my background is agriculture. So it's very interesting how you study the Plasmodesmata and you found that funnel Plasmodesmata, the number of funnel Plasmodesmata will increase the phloem unloading. Is that correct? So my question is that can that characteristic be engineered? So as you said that if, for example, apple trees can have more funnel Plasmodesmata, then it will be inducible for more phloem unloading. More sugar will come to the parts where you are interested in plants.
Right. This is the aim. We currently look for the signal that triggers the formation of funnel Plasmodesmata. This is one of the key factors that we need. So in understanding what signal triggers it and how we can utilize it to direct to tissues of interest. So this is the step that we need to do in order to increase yield. Yes.
Okay. So have you gone to molecular level for that question?
Yes, we do that.
Okay.
Yeah.
Wow.
Yeah.
That will be interesting.
Yeah.
Okay. Since there's no more question being posted in the chat room, so we will then end the presentation and Q&A session for Professor. Okay, is there one?
There's another question, yes. How to
Okay.
determine the relative hydraulic pressure and determine the cell position when cutting cells with a microtome? You're absolutely right. This is impossible. We do that a different way. So we do that in situ in the plant, and we have developed a device that is called DIMP, diffusion injection micropipette, to do some of these, and we have developed a pressure probe that we can impale directly into cells, and that shows us directly the intracellular pressure. So we published a paper on this, I think in 2015, is a pressure probe. If you can find it, just send me an email. I'm happy to forward you the link. Yeah?
Okay. Is there any more question?
How to apply the results of this research to improve crop? Well, it is, first thing, this is basic research, and basic research to turn into crop yield changes or so usually takes quite a few years. And so, as I just said, the next step is to find the signals that induce funnel Plasmodesmata formation. And when we know the signal that induces funnel Plasmodesmata formation, and we can direct it to things of interest, like in a carrot, we would like to direct it to the root, right? In a tree, like an apple tree, we would like to direct it to the fruit, not to the root. So we need to find the signals that induce these funnel Plasmodesmata formation. That's one project we work on right now. Once we have this in hand, then we can actually improve crop yield, yes. Okay, one last question maybe. Do I have time for one more? Or is
yes, yes, one
okay,
more. I have one last question.
What tools are used to determine the pressure relative? Again, this is it's called a pressure probe. you can impale it into individual cells and you get a direct reading of the intracellular pressure. And then if you want to have relative to other cells, you can measure parallel in different cells. This is possible, and there are papers on this. Again, we published a new method on this in 2015, I believe it is. Yeah. Okay. Well, thank you very much for the invitation. I appreciate it.
Oh, thank you. So, well, on behalf of the organizing committee of IconSmart, I thank Professor Dr. Michael Knoblauch for his contribution in our conference. Such an informative and interesting talk for us. Thank you very much again, and it's our pleasure to present the certificate for you as a plenary speaker. The organizing committee will send it to you. Okay, thank you very much again, Professor Knoblauch.